Hello, everybody. We are now on chapter 3, cell biology. And so we've gotten through the first two chapters of Unit 1. We're at the last chapter. So make sure you're looking at chapter 1, 2, and 3 to prepare for your Unit 1 exam. And so today we're going to talk about the cell, of course. And so the first thing that we will do is go through all of our different organelles of the cell. And so think about our organs and organ systems in the body. They each have their own job, and eventually they all work together to allow us to function as an organism. Think of all these organelles as organs of the cell. So all of these organelles are going to have their own individual role and function, and in the grand scheme, they all work together as a system to allow that cell to function. Okay, so before we even hop into this chart on the left side, I want to start going over a few that aren't on the chart that are just as important. So, arguably, the most important part of our cell is the nucleus here. And the reason I say that is because our nucleus is what is going to contain our genetic information in the form of DNA. And so without our nucleus, without our DNA, it's kind of like we don't have the recipe to make our dish. We have to have that recipe. We have to have that genetic information to be able to replicate and make more cells. This pink on the very interior of our nucleus, we call the nucleolus. And then this purple outer layer here is what is known as the nuclear envelope. And on the nuclear envelope, you can see we have these holes, or nuclear pores is really the proper term there. And so that's there so that we can get material both into and out of our nucleus. And you'll hear that nuclear envelope word later on in our lecture when we talk about the cell cycle and mitosis. I also want to point out this yellow substance here. This yellow kind of jelly-like substance is going to be our cytoplasm. And so cytoplasm is kind of what holds our organelles in place and gives our cells a little more support and structure, but it's also our liquid matrix on the interior of our cell. So it's going to have a lot of ions and nutrients and other substances that are either dissolved within the cytoplasm or traveling through the cytoplasm. The blue on this outer portion is what we call the plasma membrane or cell membrane. And we'll talk about this more in depth in a few slides. And part of our lecture today, we'll talk about how we get material across this membrane, both into and out of the cell. Now we can jump over to our chart here, and we'll start with our first two, the endoplasmic reticulum. So we have a smooth endoplasmic reticulum and a rough endoplasmic reticulum. Sometimes you will see those just abbreviated as smooth ER or rough ER. And they're housed here very close to the nucleus. So our rough ER has these dots known as ribosomes that we'll talk about here in a second. Our smooth ER does not have anything on the surface of its membrane. So our smooth ER is responsible for lipid synthesis, the synthesis of fats within the cell. Before we talk about the rough endoplasmic reticulum, I want to point out what these dots actually are, because they're not just dots. They're actually another organelle known as a ribosome. And so our ribosomes are the protein factories in our cell. These are the organelle that are going to get the instructions from the DNA, and we'll talk about this a little bit later. And they're going to actually use those instructions to make proteins. And so you can see we have ribosomes on the surface of our rough endoplasmic reticulum. And we can also find ribosomes just floating within the cytoplasm. Those are known as free ribosomes. So those are our protein factories. And so our rough ER has ribosomes all over it. So of kind of makes sense that our rough endoplasmic reticulum is responsible for protein synthesis. So our ribosomes are initially responsible for creating that chain of amino acids. Our rough endoplasmic reticulum is going to kind of do a little bit more to modify that protein and package it after the ribosome has actually created it. So ribosomes are forming proteins. Rough ER is going to then modify and synthesize those proteins so they're ready to go elsewhere in the cell. Our next one here is the Golgi apparatus. So that's this guy right here. You can see it's kind of a similar membrane structure to the endoplasmic reticuli we just talked about. And I like to refer to our Golgi apparatus as the mailman because its job is to kind of pretty up our package, modify it a little bit further, and then transport it wherever it needs to go. So protein modification and transport. Our rough ER is kind of like FedEx or UPS. It's doing the first part of the work. It's doing the initial packaging after it leaves the factory, and then it's delivering it to our Golgi. He's your local mailman that's going to actually bring it to your house. These orange ovals here are what we call our mitochondria. And so they're the powerhouse of the cell. They're our power plant. That's how our cell is going to produce energy. And when we say produce energy, and we're referring to cells, we're always referring to ATP. So we saw that molecule earlier in this unit, and our mitochondria are the ones that are responsible for producing this so that our cells have energy to function. And so it makes sense that cells that would use up more energy or need energy more often are going to have a higher concentration or more mitochondria. One of the cell types that's going to have a lot of mitochondria, if not the most, is going to be our muscle cells. We talked about how our muscular system is always working, and especially something like our heart, which is a specialized type of muscle, really is working at all times. So we're always going to need energy within our muscle cells, so they have a lot of mitochondria. Our next one is a lysosome, and so that's this bubble-like structure here. And so within this lysosome, it has digestive enzymes. So its job is to break apart different molecules. And so these digestive enzymes aren't necessarily going to break it down into waste all the time or break it down with the goal of getting rid of something. Oftentimes, when we're trying to break apart molecules, we're trying to recycle the component parts to utilize again. It's much more efficient if we can recycle some of our pieces to make something new. And so think back to chapter 2 when we talked about carbohydrates and proteins and lipids and nucleic acids. For most of those categories, we said there was a building block. So for carbs, it was our monosaccharide. For proteins, it was our amino acid. And for nucleic acids, it was our nucleotides. So rather than destroying a giant protein, our lysosomes can ingest it and break down those bonds so that we have just amino acid building blocks again that we can now utilize to make a new protein. Our last two on the list are relatively the same structure, but they're different based on where they're going to go. So our first one, a phagocytic vesicle, is going to ingest molecules. So first we have to figure out what is a vesicle. A vesicle is like a bubble. So a lysosome could be considered a special type of vesicle. And these bubbles from our Golgi apparatus would also be considered vesicles. And so vesicles are little bubbles, and it's often how we're going to transport substances across the cell, whether it's in the cell going to a separate structure or whether we're taking it out of the cell. So for a phagocytic vesicle, phagocytic means it can ingest material. It's going to bring something into the cell. It's ingesting molecules. A secretory vesicle, like this one up here is showing, is going to secrete molecules. So sometimes we'll use a vesicle to bring something in. Sometimes we'll use a vesicle to secrete something out. And what these bubbles are, they don't just kind of appear out of thin air. They're pinches or little blebs that come off of our different membranous organelles. The Golgi is really the best example of this, and even parts of our cell membrane have the capability of forming these vesicles. Speaking of our plasma membrane, that's what we're going to talk about next. And so this first slide just gives you kind of a zoomed in picture of what our actual structures are that we find within the plasma membrane. And so the most important characteristic of our plasma membrane is that it is selectively permeable. And so a selectively permeable membrane is going to let certain things in and out, but other things it's going to make sure stay in, other things it's going to make sure stay out. It's the way that we can control what is coming into and out of our cell. If it just let everything in, we'd have problems. If it didn't let anything in, we'd also have problems. So we have to select what we want to go in and out. And the main thing that gives it that capability or that characteristic is this phospholipid bilayer. So we introduced phospholipids last chapter. And so you can see we have heads and tails of phospholipids, and we call it a bilayer because we have one layer facing the inner membrane surface or the intracellular fluid, which has the heads facing in the inside, tails towards the middle. Our external layer has heads facing the outside or facing the extracellular fluid, and again, tails towards the middle. So they're organized tail to tail. I'm going to pop over here because we introduced these words in the chapter 2 lecture as well. So our tails are hydrophobic. They're nonpolar. They do not interact with water well. And so these tails not wanting to interact with either the extracellular fluid or intracellular fluid make this a really, really good seal. So things aren't going to get across real easily. Our heads are hydrophilic, meaning they love water. They're polar, just like water, so they're going to interact well. And it's important that it's okay with interacting because our intracellular fluid and extracellular fluid are going to have a lot of water in them. A couple other things to point out here. We have two different types of membrane proteins. And so I'm looking right here. You can see this purple one that's labeled an integral protein goes all the way through the membrane. So part of the protein comes out of the external environment, part of the protein reaches all the way to the internal environment. A peripheral membrane protein is going to be a protein that is on the periphery, just on the extracellular surface. You can see this one's an integral protein again because it's going all the way into this layer. And then this one we would call an uh, internal membrane protein versus a peripheral because it's at the internal environment. These yellow chains are carbohydrates that have a few different functions, but mostly they're there for identification. So it's the way of the cell telling other cells around it, what type of cell am I? What do I have going on? All right, we talked about some of this already. So the main job is to define and separate that inner and external environment. We want to keep what's supposed to be outside outside, keep what's supposed to be inside inside. Our water-soluble molecules are going to have a hard time passing through without having extra help. We'll talk about this here shortly. So think water and fats don't mix very well. Our cell membranes are a huge double layer of fats, so it makes sense that a molecule that's water soluble isn't necessarily going to just pass through. An important structure along with those phospholipids and proteins that we haven't talked about yet is cholesterol. And cholesterol is needed for us to maintain our proper membrane structure. So this is really important for our membrane kind of staying together and staying in that organization of that phospholipid bilayer. So I said this as well. It's sometimes called the fluid mosaic model because it has that wave-like appearance. So our two layers are going to kind of flow together. It's not a really rigid wall. We talked about peripheral and integral proteins. those receptors or some of those carbohydrate chains, that allows it to um, identify itself. And it's also going to have receptors for things like hormones or other chemicals that when that chemical or hormone lands on its receptor on the cell, it causes some change in that cell to happen. So it has receptors based on different hormones or chemicals that might kind of give our cell instructions on what to do. Our proteins often function as channels and or pumps, which are going to help us control what substances can enter or exit our cell. And certain substances, like we said, are water soluble substances. They're not going to be able to make it in anyway. So we have to have a channel or something that allows them to pass through that membrane. So the first type of transport we'll talk about is active transport. And the best example of this is our sodium potassium pump. And so in general, things like to flow from an area of high concentration to an area of low concentration. So think of us going from high concentration to low concentration as flowing with the current. So think of a river. High to low is going to flow down. We want to naturally flow down what we call our concentration gradient. So things will naturally go from high concentration to low concentration. In our example here, and in most active transport examples, we're trying to go against that concentration gradient. Rather than naturally flowing with the current from high to low, we're trying to swim against the current, and we're trying to go from a low concentration to a high concentration. And so when we want to do that, because we're fighting against how our material naturally wants to move, it's going to take energy. So our example here is our sodium potassium pump, which is super important for our nerves to maintain what we call their resting potential. It's what allows our nerves to maintain that ready potential of firing. And so in our nerve cells, when we're not firing them, we have a lot of sodium on the outside and a lot of potassium on the inside. And so our pump is continuing to pump more sodium out where we already have a lot of sodium, and it's bringing potassium in where we already have a lot of potassium. So we're pumping both sodium and potassium against their concentration gradient. We're taking an area that already doesn't have a high concentration of sodium and pumping it to an area that already has a really high concentration of sodium. We're taking an area of potassium that has a high concentration of potassium, and we're bringing more potassium in. So we're going against that natural flow. And so our primary active transport example here, we're utilizing ATP. So ATP hydrolysis, if you remember that from our last chapter. So hydrolysis to break that bond between our three phosphate groups. So we're going to break a phosphate group off so we have energy available, and that's going to power this pump to pump sodium into a higher concentration, pump potassium where there's already a high concentration. Secondary active transport is almost like borrowing energy. So this time we have primary. We need ATP to make this pump go. In this secondary example, we're using energy that is created by this movement of ions, and that also allows us to bring in glucose. So this is a combo method. So we're really getting the energy from sodium rushing in, but that also allows us to bring glucose in. So this example, we need ATP. Primary active, we have to have ATP. Secondary active is usually a byproduct or something else that happens along with this primary that allows us to do something utilizing that extra energy. Okay, now we're going to talk about passive transport. So passive means it's not going to require energy. So diffusion is the primary one that we talk about, and then we'll also talk about osmosis. So diffusion sometimes can happen just across the membrane, depending on what our material is. An example of this is oxygen and carbon dioxide. They naturally go across our capillary membrane. We don't need a protein to transport them. So remember what we said just a moments ago. Things naturally like to go from a high concentration to a low concentration. And so in this case, we just said we have a whole lot of potassium in our cell, so it's going to want to go out. We have a whole lot of sodium that's outside of our cell, so it's going to want to come in. And so we have different types of channels. This one here, this purple one for potassium, is called a leak channel because it stays open, so potassium just kind of slowly leaks out. This is called ligand gated, meaning it needs something to land on its receptor to tell it to open. So here we see we get our ligands bound, our receptors are now filled. So now our gate opens. Sodium is high concentration here, low concentration here. So it wants to travel through. So no energy, and we're going from high concentration to low concentration. So don't get it confused that even though we're using a channel protein here, it doesn't take us energy to utilize that protein. It's not pumping like our sodium potassium pump in our last example. So osmosis is referring to the diffusion of water. And so in this case, instead of the substance moving from higher concentration to lower concentration, the water is going to move in an effort to try to balance those concentrations out. And so we'll talk about three different types of solutions to illustrate how osmosis functions. So the first one we'll talk about is this center one because it's the easiest to start with. So we have our red blood cell. So we're taking it out of its normal environment and we're placing it in an isotonic solution. And so we see here isotonic means it's an equal salt concentration. So the water has the same amount of salt as our red blood cell does on its internal environment. So the whole goal of diffusion, remember, is to balance out concentrations. When we have a concentration gradient or a difference in concentrations, that's what is going to drive the movement. In an isotonic solution, there is no concentration gradient. There's no difference in concentrations, so the water isn't going to go anywhere. Our cell is going to stay exactly the same. In this case, we're putting our red blood cell into a solution that has a low salt concentration. So our red blood cell by default has a higher concentration of salt on the interior of the cell compared to the water. So we can't go from salt, from that higher salt from the cell into the water. So the water has to move into our cell. And so that's going to cause our cell to expand. And so if our cell expands too much, it can burst, which we call cell lysis. Or if a cell lyses, that means it ruptured or was destroyed. So because we have less salt in the water than in the cell, the water is going to move into the cell to try to dilute the sodium, and that's obviously going to cause that swelling. So it's not always going to lyse. That only happens if too much water goes in, but we'll always get that cellular swelling. So think hypotonic, it's going to be a big O. Our cell turns into a big balloon. For hypertonic, the way I remember this one is that you want hyper kids to go outside. So this time our water or our solution, it has a higher salt concentration than our cell. So when we put that red blood cell in, the water is going to go out of the cell to try to dilute our solution. And so we call this shrinking or this kind of crumpling of our cell, a cell crenating or crenation. So lysis, if we swell and rupture, crenate if we're kind of crunching or shrinking. All right, so we talked about the cell cycle in lab. So I'm not going to spend a really long time on this, but we will go through some of the key pieces really quickly. So the cell cycle itself refers to this entire cycle. So the cell cycle is this entire cycle. We talked a lot about mitosis, which remember is just one small wedge. And you'll see our diagram in our lab manual is just a little bit different. We have another wedge that has cytokinesis, but in this diagram, it shows it as kind of exiting the cycle. That's when we have two new cells. So let's talk about it real quick. So G1 is our normal cell, routine metabolism. So our cell is just going along and trying to grow. In S phase, we're going to actually get DNA replication. So we're going from one copy to two copy in preparation to be able to divide. Our G2 or growth 2 phase is really when we're making our final preparations for cell division. We're making sure we have everything ready to go before we actually enter our M phase or mitotic phase. And so remember all of this majority of our circle here is interphase. Interphase. So when you look under a microscope, you're going to find a whole lot of cells that are in interphase because it's a much larger proportion of our cell cycle than our actual mitosis or division phase. And then remember, cytokinesis is when we're going to actually get the split. So the cytoplasm is going to divide and separate into two new cells. All right, so we split mitosis into four phases. So now we're zooming in on our four actual phases. And so in interphase, we don't really have a whole lot going on that we can point to. You can see in the nucleus, we have our chromosomes are not organized yet. We call the thread substance of DNA. This is how it normally hangs out. That's known as chromatin. So this is our thread. And then we first see our centrioles pop up, which will have a role later on. So now we're actually entering mitosis. This is our first phase of mitosis with prophase. And so now instead of just being thread, we're actually organized into chromosomes. And so each half of our chromosome is known as a chromatid. So the material itself, our way DNA hangs out unorganized, is chromatin. Once we organize, we have a chromosome made up of two identical chromatids. The center here we call the centromere, and that's where we're going to actually attach to be pulled apart. And you can see our centrioles are starting to produce their spindle fibers that will eventually attach at those centromeres, or the other word you'll see is kinetic core. Some will break it into pro-metaphase, or you could call it, say, early prophase, late prophase, because pro-metaphase basically just means we're in between prophase and metaphase. And so now we can see our nuclear envelope, our surrounding of the nucleus is gone. We can see our chromosomes, and these spindle fibers are starting to attach to the chromosomes. The difference between prometaphase or late prophase and metaphase is that in metaphase, our chromosomes are going to line up directly along the equator or middle of the cell. So this is one of the easier ones to identify because you can see them lined up across the middle. There's one centriole, two centrioles, and these are the fibers that are running, the spindle fibers that are connecting to each half, to each chromatid. Up here, you can see we still have kind of very unorganized and yarn. That's our chromatin. In prophase, it actually organizes into chromosomes. Prometaphase, we have our chromosomes starting to organize. They're getting towards the middle, and then it's not metaphase until we're all the way lined up across that equator, we call that. All right. Anaphase, we know, starts once we actually have that separation. So this is another one sometimes we can split into early and late because we can see they just split here when they're getting closer to their edges of the new cell or in the centrioles. We know it's later anaphase. So you can see now they're separated. We can see that space in between. And so we have identical chromosomes now. They're not chromatids anymore because they're separated. So we have identical chromosomes that are traveling to either pole of our new cells. You can start to begin to see the very beginnings of this cleavage furrow in anaphase, but it's really telophase where this emerges. So we can start to see where the cytoplasm is beginning to pinch off. So we now have our nuclear envelope back on either half. So our chromosomes are back surrounded by the nuclear envelope. Our spindle fibers are gone because we're all the way separated. Their job is over. And we're starting to get our cleavage furrow where eventually cytokinesis will complete and we'll get two brand new cells. So these we call two daughter cells. And these are completely identical. So completely identical because we copied our chromosomes and we split them directly in half. So two identical cells. And so cytokinesis, like we showed in here, is not technically part of the mitotic phase. That's kind of a separate process that happens after mitosis. So telophase is where mitosis or that M phase ends. And then once that cytokinesis happens, both those cells are going to be back into this G1 or interphase. So they're really, I would say, just try to remember some of those key characteristics. So interphase, we're not organized into chromosomes at all. Prophase, we can see that chromosomal organization, and we can see our centrioles are starting to produce spindle fibers. We're still in prophase even when that nuclear envelope dissolves, and we're starting to organize further and attach our spindle fibers. Metaphase doesn't truly become metaphase until we're all the way in the middle. And so now our spindle fibers are all attached at the centromeres, and we're going to be able to separate those chromosomes directly in half. Anaphase, we're now fully separated. So if you still see them connected, that's metaphase. This is anaphase. Telophase, we get our nuclear envelope back on either half, and we start to see the cleavage furrow that is going to eventually continue. And once they're fully separated, that's when we say cytokinesis is complete. Okay, last concept for today, the last few slides here. We're going to talk about transcription and translation. And the big things you guys need to know about this is what structures are involved and kind of where it happens. So transcription is in the nucleus. So this is going to happen in the nucleus. We said DNA is kind of our recipe. It's our master copy. And so we don't want to just send our master copy floating around willy-nilly. So we use mRNA. This M stands for messenger RNA. And it's going to actually make a copy of our DNA template that can then be used by our ribosomes or eventually used by our ribosomes. So transcription is going from DNA to produce mRNA, and it's happening in the nucleus. So there's a reason that's underlined. Translation is going to happen within our ribosomes. So this could be a free ribosome or it could be one of those ones that we saw on the membrane surface of our rough endoplasmic reticulum. And its job is to actually cause this protein production. And so it's going to use that mRNA kind of template. We're also going to use tRNA. So we'll talk about that in a second. and we're going to actually produce our protein in the cytoplasm. In the cytoplasm. So transcription in the nucleus, translation in the cytoplasm, and we'll go through our steps. So we're going to keep it really simple for transcription and translation. So another reminder, this is happening within our nucleus because that's the only place that we're going to find our DNA. And so initially, DNA starts off as what we call a double helix structure, that double ladder. And so the first step is that we need half of our DNA to unwind. We need to make one strand available. This is when our RNA is going to make a copy of the strand. So this shows our purple DNA unzipping and our gray RNA coming in to match up with the pairs. So for RNA, U and A are a match, C and G are a match. In DNA, C and G are still a match, but A matches with T, and this is what is being shown up here. So for DNA, our first four, cytosine, guanine, thymine, adenine are our nucleotide pairs. So A goes with T, G goes with C. That's for DNA. For RNA, we have cytosine and guanine are still pairs, but RNA doesn't have thymine. So adenine pairs with uracil. So A goes with U. And so this is just making our mRNA. So you see this down here. Our mRNA is forming, and we're making a copy because we know C goes with G, U goes with A goes with T. In this case, we have the A, C with G, C with G, U with A. So we know what is going to pair with this. This is giving us what our DNA is showing. Step number 3 is our RNA actually going from RNA to mRNA. So it goes through this 3-step process. So splicing deletes an extra RNA. So if we have something that's just a placeholder, now we're going to get rid of that because it's just holding a place. It's not actually having any code for our protein. So it's unnecessary. We're going to get rid of that now. We're also going to add a cap and a tail. And what the cap and tail do, more or less, is tell us that that's where we're starting and that's where we're finishing. So the cap and tail is really just our start and our finish. mRNA is then going to leave the nucleus because now it's ready to find a ribosome that can actually produce the protein. Remember, this is going to occur in the cytoplasm, and namely our ribosomes. This is when we bring tRNA in, or transfer RNA. It's going to bind to an amino acid. So this is one example. Remember, we have 20 different amino acids. So tRNA now is going to bind with an amino acid. This tRNA is going to find its codon on the mRNA. And so we call this recipe or this pattern on the tRNA an anticodon, and it's trying to match up with its codon on the mRNA. And the same rules apply. A and U combine still or make a partner. G and C make a partner. And so for our proteins, we kind of know the amino acid that's the next building block based on these 3 nucleotide sequences. So that's why we can see 3 in a row. Okay, so step 3, tRNA is now going to bring that amino acid into the ribosome. So step 1, our ribosome, our mRNA, and our tRNA are going to combine. So now, even though this is 1, I'm just trying to make sure you're not confused. We're really here. And so this is a little more detailed than you need to know, but once our tRNA with its amino acid enters the ribosome is going to create the peptide bond between our amino acid blocks, And depending on how long of a chain we're creating, we're going to produce a protein. So proteins can be a few amino acids strung together, but oftentimes they are thousands of amino acids strung together. And that's why we have that cap and tail, because we need to tell our ribosome when to start and stop. Okay, and so that gets us all the way through chapter 3. So for transcription and translation, I really want you guys to keep it as simple as you can. Just stick with our steps here. We tried to simplify it as much as possible. The diagrams are there and illustrations to help you understand it. But if there's more detail in our diagram that's not here in our steps, then just kind of keep it as nice to know information. And please reach out to your instructor with any questions for Unit 1. Thank you.